Okay guys, we're back and we are now going to take a look at the DNA and its role in the production of proteins. So we're not going to use the DNA, the original DNA molecule, to make more DNA through DNA replication. We're going to use the DNA to produce proteins using the DNA as the code. All right, so here we have an original piece of DNA. Again, we've got this idea where you'd have this 5' to 3' going on, right? And these are anti-parallel strands. And let's say that we're going to use this upper blue strand. So this blue strand right here is a gene, okay? So in order to read this gene, well, we need to start similarly to what you see when we need to replicate the DNA. We need to tear the two strands apart so we can get at that blue strand as a gene and read it, okay? Now again, we want to read always making a new strand from 5' to 3' direction, okay? So if we pull this apart, okay, so now we have the blue strand, we want to read starting on the 3' end of that gene, making a new 5' to 3' strand, but now we're going to produce not DNA, but something called RNA, right? So, but it's very similar, right? So if we just ignore the gray for now, because that's not the gene in this scenario, we're going to focus on the blue, okay? Every thymine is going to bind to an adenosine, right? So if we had a T and an A binding up, that makes sense. And then we have an A and a T binding up, but what do we see here? We see actually a U for uracil. So this is one slight difference between DNA and RNA. Again, they both end in NA for their abbreviations, right? So they're both nucleic acids with nucleotides as their subunit, as their building block, but they're slightly different dialects. Think of it that way. They're the same language. We're not changing the language here. It's still nucleotide language. But we have uracils instead of thymines, okay? So we have not a TG, we have a UG, and so on. Anywhere where there were T's in the complementary strand of the original piece of DNA, we now have U's, okay? So now we have made a transcript, an RNA transcript, okay, versus the DNA we had earlier. So here we have our RNA, and this RNA is now almost like some kind of notes from the code, right? We've taken some sequence from that code, and now we're going to use that code to make something useful. Just reading a book about how to build a shed doesn't make a shed, right? So here we've read the book, we have notes on the blueprint as to how to build this thing, and now we're going to actually produce proteins from this RNA transcript, right? Now, it used to be that a lot of scientists thought that DNA and RNA, these that we know to be the genetic material, our code for life today, well, we used to think it was protein, but now we understand that every 3 RNA nucleotides, okay, those 3 code for a particular amino acid. So it takes 3 nucleotide in sequence to code for a particular amino acid, and so we see a few sets of 3 here, each one of them coding for a particular amino acid, okay? And we're going to go over this as something you're going to want to be able to do in the exam, to be able to take a sequence and not only do DNA replication with it, like we talked about last clip, but do some transcription, is what we call this, going from the original DNA code to an RNA transcript, okay? And what we call this when we go from the transcript to the actual protein, we call that translation. We're changing our language at that point, okay? So we have transcription up here, going from DNA to RNA. We have translation down here going from RNA to protein, and this is generally a lot of times called the blueprint, or the central dogma is what they call it. This idea that DNA codes for RNA through transcription and RNA codes for protein through translation. So DNA to RNA transcription, we're taking notes, same language though, and next step, RNA to protein, we have a message in the RNA, so M for messenger RNA, and protein is produced from that. And this would be a functional protein. It may be an enzyme, for example. All right, here we have an image of how this transcription, this first phase, might take place. In blue, again, this is similar to what we just saw, but it's just slightly different rendering, okay? In blue, I see here on either side our original DNA. One side of this DNA, the bottom part down here, okay, is serving as a gene, okay? And it's serving as a gene starting right over here where we see our 3', okay? Now, we talked about initiation, elongation, and termination when we were talking about DNA replication, and here we see this again, all right? Here I see the idea of initiation, elongation, and termination. However, here it's not DNA polymerase involved, it's RNA polymerase. So initiation is RNA polymerase will come in and lock on. Okay, so where's it going to lock on? Well, it's going to lock on over here where the gene begins. Okay, then it's going to ratchet its way across. It's going to work its way along those nucleotides just like DNA polymerase did. As it does, it's going to place AC's U's and G's and base pair them with the AC's T's and G's we see in the original parent DNA strand. And so here you see that happening, right? Here's our RNA being produced. It started with a 5' end, building from a 5' end, and we see U's instead of T's. Ultimately, this will come to an end. There will be a point where this will stop, and we'll have termination, and the RNA polymerase will break away. So here's that RNA polymerase elongating the transcript, and then ultimately it will break away. We don't see that in this image, but ultimately it will come to an end where it terminates that process. Now, the next step we would need is the translation process. We have the RNA transcript. This would come off of that DNA. DNA would go right back together the way it was, right? We're not destroying the DNA. We're just taking notes from that code. And here we would see a rendition of that transcript. This is our mRNA, okay? And that mRNA is going to serve as our code, okay? We would start on the 5' end of that RNA code with a ribosome attaching, and it would ratchet across, reading every 3 of those RNA nucleotides and bringing in an amino acid for each of those 3. Each set of 3 RNA nucleotides will code for 1 amino acid. And ultimately those amino acids are strung together. The ribosome detaches, and the whole translation process ends. Again, we see these words: initiation, elongation, and termination. So we want to make sure we understand what are these phases during DNA replication, what are those phases during transcription, and what are those phases during translation. So in this case, it's not an RNA polymerase grabbing on, elongating, and coming apart. What we now have is a ribosome, this protein factory, latching on to the transcript, elongating the amino acids coming together to make a protein, and then detaching when that protein is finished. Okay? Now what we'll want to be able to do, what you'll want to be able to do during a test, is to read that code, be able to tell me what are the particular amino acids that are coded for throughout that transcript. Now I'm not going to expect you to memorize this chart. That's ridiculous, but I will give you that chart, okay? And you want to be able to use that chart to read a transcript, okay? And give me the amino acids that would string together. So let me give you kind of an example. So here we have that chart, right? And imagine now I've given you this particular DNA template. This is our gene, okay? Okay, so this is our gene. This is the side of the double-stranded DNA that we're going to read at this moment, okay? The other side might be a gene for something else, but we're not going to focus on that side. We're going to look at this side, okay? So here we have our 5' and our 3' end, right? And we want to start building always with a 5'. So we want to find on our opposite side the 3' end and start building with the 5' first as we go across, right? So if I tell you that this template is going to be serving as a template as a gene, or I say we're going to want to do now RNA transcription or something like that, we know we're not talking DNA replication anymore, right? Any mention of a gene, any mention of a protein being produced, or transcription means we're going to use this to produce a useful product, not just to copy the DNA. So what we would do is we'd have an RNA complement, a transcript being produced. We would have to start on this end of our DNA, our gene, right? Because we want to build starting with a 5' first and go that way, right? So the RNA polymerase would latch on over here on this side of the gene, and it would place A's for T's, U's across from an A, right? We don't have T's anymore with the RNA transcript. G's for C's, C's for G's, and so on. We're working our way across, okay? Now, if I pose to you the question, okay, from this gene, what will the sequence of amino acids be? What I would then do is I would take my transcript, take my RNA, this is our mRNA transcript, and I'd use the chart. Okay, so the first letter of this particular sequence is an A. So I'm going to find my A's, my first letter A. Okay? I'm going to find my U, that's my second letter, okay? So second letter up here, I'm going to find U. So I'm basically now I'm limited to this particular box, right? And then I'm going to go to my third nucleotide, which is a G. So here we found it, right? Here we have AUG. That is coding for a singular amino acid called methionine. And I don't need you to know the full name, but MET, this is our start amino acid. Okay? We go to our next one. Let's try the next one. CCC. Well, I have my first letter C. I've got my second letter C. I got my third letter C. We're talking this one right here. This has got to be proline. This is our second amino acid. So you get the idea, right? Let's do the next one. GGA. Okay, I've got G's for the first one here. G's. So we're somewhere down here in this box. And then we got A, right, for the third one. So this one is a glycine. Okay, so you would just write, you indicate that GLY, that's our third amino acid. Now let's try this last one here, okay? This last one, UGA, let's find that. U, first letter, G, second letter. So we're up in this area somewhere, and we have A. Check that out. This says stop next to it. It's not actually going to code for anything. This is an indication that the ribosome needs to stop what it's doing. Okay, so it started with a start methionine, and it's going to end with a stop signal like this. Okay, it doesn't actually code for anything as far as an amino acid. We'd end up with methionine, proline, glycine. This is our amino acid sequence. Okay, so what we're doing is we're using these three nucleotide sequences and finding out the amino acid sequence. When we look at this sequence in the DNA, we usually just say it's a triplet. Okay, so we have a triplet of DNA within our gene. When we look at the three nucleotide sequence within RNA, we call it a codon. It's coding for something, so it's a good name, right? It's coding for a particular amino acid based on that 3 nucleotide sequence. All right, so let's call that quits for now, guys, and we'll launch into our next section soon enough. Okay.